Good evening, everyone. Nice of you to come to this instead of going to dinner. I'm honored. And Beech Leaf Disease is still the most potent and terrifying new disease we've got. So it's one that you do want to keep up with. I'm really glad you're interested in the education on the topic. It's also one of these things that I haven't seen since dogwood anthracnose. We've got one disease that's threatening our native trees as well as threatening our ornamental types. And that makes it really important to me in my day job, which is concerned with protecting ornamentals in their production or their display. And also my personal affiliation with the woods. I want the trees to be healthy in our woodlands. And beech is suffering in both locations right now. So amazingly, the American beech has a gigantic host range. I had never been aware of this, but it's practically the east half of the United States. And Fagus grandifolia is our tree, and it's probably best known for people carving their initials into it, but it's been a really good support for wildlife as well. It's a very giving tree, just not the world's most famous lumber species. The thing which is now assailing it is a nematode, which you might not have really known much about before if you were an arborist because they don't usually figure in arboriculture. The one that's causing this problem was only discovered a few decades ago. It's called Lidolencus, and the particular specific one is Crenati Forma Specialis mecanii. And there's a lot of meaning in that name because the Lidolencus is named after a Dr. Lee, who was the person who was very important in developing the identification for the nematode, which was a new type of nematode. Crenati refers to the species name of the Japanese beech, which we only have here in Arboreta. And then mecanii is our American specific strain because that's the name of the man who was clever enough to figure out that this disease was being caused by a nematode. It took us 6 years from the first notice of symptoms to making the association with the nematode. And he did it, so we owe him a lot of thanks. This is what that nematode looks like. Up at the upper left, you've got a female nematode, and at the right, you have a male Lidolencus. And then the one down on the bottom, I'm not sure what its sex is, but it looks terribly comfortable inside the leaf, sitting on a batch of leaf tissue there, a batch of cells. The states that are affected up till now are listed here. And then there's also Ontario in the mix. And that's one of the reasons why we know that this is a disease that's going to do well, even if we're having a cold winter, because the parts of Ontario where it's been seen are colder than we are, by far. This is the map from 2023, and there's been a little bit of growth since then, but this gives you a bit of an idea. If you look over to Ohio, you see one little spot that's white. This is the county, Lake County, where the disease was first noticed. And it began spreading from there. It actually got into the southwest tip of New York State first, but then fairly early in the process of things, in 2019, it leapt over to Long Island and got into the southern Hudson Valley, which is in this sort of navy coloring. And it's been moving ever since. The moving is, to some degree, a question of when people had heard of it and really bothered to look for it. But I think there really was a speedier appearance of symptoms here on the coast than there was in the central parts of New York State. There's something about nearness to water that seems to really encourage the disease, and that's probably something about high humidity. But it'll take more research before we really know. Moving to Europe, and this is a really funny map. I copied it from the web, and for the strangest reason that I can't comprehend, New York is on the map of the European beech natural range. Somebody must have been talking about the New York Botanical Garden or something. But at any rate, European beech is really important in Europe and Scandinavia. It's a very it's a tree that's integrally linked into their history as peoples, and it's widespread. So one of my biggest fears is that the disease that we have here in this country and that we have in Japan will make its way into Europe because we can see symptoms on our European beech here. We know it's not immune in any way. There might be some immunity in the population, but it should be pretty much as susceptible as the American beech if it appears there. So that's of great concern. And beech goes way back in their history because the Old English word for beech is bok, which is what our word book comes from, because it was used for the paper that made the books. Okay, did you get to see this slide? It's a change of slides. There are several things that we knew about beech already. When this new disease came on, there were things that we had seen in other parts of New England and New York. Up at the upper left-hand corner is a pretty familiar one. If you ever go hiking in the Catskills, you'll certainly see lots of this, which is the beech bark disease. That's probably why this one is called beech leaf disease, because they were trying to make something parallel to it, but different. And the beech bark disease is a fungal infection. You're looking at the sexual spore cases of the pseudonectria that causes that disease, but it can only infect a tree if first a scale insect has worked over the bark and created nooks and crannies to help the fungus get established. So it's a fungus plus insect problem. And down at the bottom, that's not a beech tree, but armillaria is one of the things that will go after beeches and cause root rot, particularly following drought stress. And then we also have the xylosandrus, just to the right of the armillaria mushrooms. If you look on the right, you can see what look like little tubes being extruded from the side of that trunk. Those are evidence of a ambrosia beetle feeding inside the wood. And their frass gets pushed out behind them, and until you have a rainstorm, you can see those little, they call them toothpicks, but they're usually sort of curly for toothpicks. So that has been seen in our part of the world, at least, causing a lot of branch dieback in beech, again, usually following a droughty period, which makes the trees more attractive to the ambrosia beetles. And then there are a couple of different kinds of aphid that can affect beech. The famous ones are the beech blight aphid that dance up and down, and this is another beech aphid that just sits there, but it's enough to cause treatments on European beech in some of the more carefully maintained landscapes here on Long Island. And then at the far right, you have one which is a fungal rot that really disturbs the integrity of the tree so that it will fall down if you don't cut it down. And that's a fungus known as Kretschmaria adusta, which is, we'll give you that as a spelling question by the end of the program. But do notice that drought is important for many of these things. Beech are fairly shallow rooted. And so for the previous problems, drought was something that added to the problem. Whereas now that we're thinking about a nematode that's in the foliage of the trees, it actually works the other way. Droughty weather seems to suppress the nematode. So the perfect weather for this problem would be to have just enough drought to hurt the nematodes, but not enough to hurt the beech. And I don't know how we define that, but that would solve the problem if we could get there. So this beech leaf disease that we're talking about tonight, known as BLD, was seen for the first time in Ohio in 2012 when John Pogachnik took a walk in the woods. And he was with Lake Metro Parks, and that's a group that has done a lot of the research on the disease since then. And now it's expanded way beyond that territory. And as I mentioned before, Dave McCann, who's with the Ohio Department of Ag, was the one who figured out that it was a nematode behind the symptoms and sent them to USDA Beltsville to be identified by Lynn Carta and the people in her group. The leaf that you see at the lower right is the first one that was given to us to identify a problem on Long Island. And that was only 1 year after this was found to be a nematode problem. In 2019, Evan Dachau of Jolly Green Tree, came to us with these funny symptoms he was seeing in the Stony Brook area. And it turned out to be our own little case of this problem in the year 2019. So you've got a number of hosts known for this: American and European beech, the Oriental beech, the Chinese beech, as well as the Crenata, which is the Japanese beech. And it's causing death of trees wherever it's found, especially younger trees. So the older trees seem to hang on for a while, and I think if environmental conditions aren't too harmful to them, they might even be able to weather this problem. We haven't really watched it for very long, so we don't know the details of survival. There is some interesting information now in a few papers that have come out of Japan that talk about the problem. So if any of you have been just waiting with bated breath to find out uh, how you would write the word nematode in Chinese characters, you can see it over here where it says senchu. That's nematode in Japanese. And they had some very excellent nematologists who started studying this disease. And they noted in Japan that there were alders and ostrias that were affected. And that has not been noticed here in this country. So I think that's an interesting question that we're not completely sure on. Are there other hosts for this? Once you start using molecular detection to find the nematode in the woodlands, you can find it in places where you're not seeing symptoms, and you can find it in places where there aren't beech. So does it have other hosts, either for just maintaining itself or other hosts that it might attack and cause harm in? But for now, it seems to be a beech disease for us. This is another map showing you the amount of the problem, all those red spots in Japan, and the larger circles there are showing you where they did molecular identifications to see that they had the same organism occurring across that whole territory. And they figure it probably started there in 2003 or even earlier. When you compare the information about the nematode that they've developed in Japan to the information about the nematode that we have here, it basically looks like the same nematode. There are just a few distinctions that have caused us to set it out as a different form of specialis, but it's by and large the same organism, genetically very similar. It has the same shape. You know, if you were looking at the nematode, you would say it was the same one. It has a lot of the same genetic material, and its host range is pretty much the same, but there's some odd differences. Our nematode doesn't actually go very strongly to the crenata that is found in Japan, and vice versa. Their crenata doesn't attack the American beech when it's being grown in Japan very well. So there are curious behavior things involved here too. But the world is concerned about this. This disease has made it onto the lists of serious invasive exotic problems that are moving around the world. And there's been some information that's been published. There's a USDA pest alert that gives you fairly recent 2024 season updating on facts about this disease. And there have been a couple of papers recently giving research on control, in particular done by the Bartlett Company that's been very involved in getting information on beech leaf disease from the beginning. This is what the nematode looks like when you have a large number of them, and it's not hard to find a large number of them if you go to the right place at the right time of year. And those funny little things that look like Vienna sausages are the eggs of the nematode. There are males and females, but they reproduce by way of eggs. And here's a close-up of the eggs. These are fairly recently laid eggs. After a while, you'll see a little nematode curled up inside that same little outside lining. This will give you a sense, you're looking at a cycle that's just written as a linear progression rather than as a circle. But if you think about overwintering, which is just about ready to start, the nematode is in the buds of the trees. This is the place where I can find it the most easily. If I want to find lots of nematodes, I can find lots of nematodes in the buds. It is not bothered by winter because that is a very sheltering place to go in the wintertime. And you will find both eggs and live adult nematodes in the buds, and they're on the inside of those bud scales, and they congregate in masses, and they feed on the tissue. So you can see the bruised tissue where they've been feeding. There's probably a temperature that's necessary for them to be feeding. So if it gets cold enough, they may go pretty inactive, but they don't die. And you can warm them up from a bud and have them squirming around in your lab pretty quickly. Then in the springtime, you get symptoms, which are familiar to, I think, 60-some% of you, where you'll see bands between the leaf veins. And those bands start out being darker than the regular leaf tissue because the beech leaf tissue is very pale when it first comes out. And those bands are showing you where nematodes are, but there's not a really high population yet in the tissue. It's actually hard to find it by dissecting out that leaf tissue. If you want to find nematodes, you wait till July, and by then they've gone through several generations of egg production and multiplication. And as you then progress into fall, the numbers just keep going up and up and up to the point that you get a population that's high enough to have a good chance of success getting into new areas in buds and making it through the winter. So there is a big difference between this disease and your average leaf spot disease. With leaf spots, if you had 10 leaf spots in the spring, you might have 50 or 200 by midsummer, because each time it rains, there can be movement of the leaf spots and propagation of the fungus into new areas. But there's only one cycle of infection for this particular pest. It's only going to be able to cause symptoms once for the year. So if you want to know whether you were effective last season doing a control action, all you have to do is look at the leaves when they unfold. It's not going to tell you better information later. You're going to find out as soon as the leaves unfold how much you were able to suppress the population of the nematodes. One of the things that the folks at USDA and David Burke at the Holden Arboretum did early on was to prove that it really was the nematode that was associated with these symptoms. And they did that by taking small plants and having them in with no leaves, just buds, and they inoculated the buds in the greenhouse in the fall. And then when those buds opened up and had new foliage in the spring, they could see those little bands between the veins. And every once in a while, when you're having a deep discussion about this, you can realize that maybe there's something else involved too. If the nematode is carrying some sort of microorganism that might be associated with the symptom development. But every time we get to listen to Paulo Vieira, who is one of the lead nematologists studying the system now, he says, hey, this nematode has everything it takes to create these symptoms. So he's not looking for anything else. He thinks the nematodes are quite sufficient to cause this disease. They're just really unusual because unlike most nematodes which affect roots or the few nematodes that actually go into foliage, this one is causing a gall in the leaves, so it's actually a leaf gall nematode, not just a leaf or foliar nematode. It causes swollen areas in the tissue. There are a lot of different Forest Service projects that are looking into beech leaf disease that we periodically get some reports on. They're concerned with how it's distributed and how it's being spread, what the vectors might be, how we detect it, how we treat it. And there are a lot of people with the Forest Service involved in that. And many collaborators work directly with them, like the folks at the Holden Arboretum and Cleveland Metro Parks. There are folks at Penn State, folks at ARS, folks at WVU, and at the Connecticut Agricultural Experiment Station. They're doing a lot of the genetic work on the disease. And in Ontario, there's a very strong research group looking into the disease. And, you know, from their perspective, really the main thing that you can do, excuse me for messing with the slides, is to just destroy the infected plants and try to keep everything healthy. When you are trying to deal with the disease on a forest basis, you have so many fewer options than when you're trying to deal with an ornamental plant. So those of you who are arborists are in the right position to do what you can with our beloved beech ornamentals to try to make things better for them with regard to this disease. It'll be much harder to do anything to help the forest population. Again, some information that Paulo Vieira put together allows us to realize that you can even look at buds from the outside and see how much nematode attack is going on inside. We didn't realize this at first, but here, for example, at the left is a healthy nematode, a healthy bud that doesn't have nematodes in it. And then with increasing amounts of injury, you have other buds with nematodes inside. This doesn't show it in this particular set of information, but here on Long Island, we often see reddening of buds from the outside if they have nematodes within. And then if you think of these different squares, rectangles here as paired, A is the same type of tissue as D, but at the bottom, you'll see how much larger the cells are. That's because of the effect of the nematodes, which are in the tissue. And the same with B, that's normal tissue at the top. E is tissue with large cells, and then C is the same thing, normal cells at the top, and then enlarged cells at the bottom. And these are all tissues from the bud scales. And this is showing you the same thing, but if you can make it out, you'll have to look closely, but the arrows are trying to help you at the left, pointing out nematodes that are coiled within the tissue. of the beech leaf, and then you see a magnification of that in the center picture where there's a long twining serpentine nematode that's feeding on the tissue. And then at the right is just a collection of the eggs of the nematode, and you can get a sense of the size of the nematode and its eggs in relation to the cells of the beech. When we look for symptoms, we look right to the buds to see what's going on. When we look in the growing season, we look for those leaf galls between the veins that I showed you, but there can be other symptoms too. There's also what they call crinkling, which is a leathery, thickened leaf that's distorted. It's usually smaller than usual, and it's rough in texture, and it's usually got a strange outline, not a typical beech leaf outline. That's, in my mind, a leaf that's just gone over completely to gall tissue. There isn't any healthy tissue within that leaf. That would be just one step short of the nematodes being there in such numbers that the bud doesn't even open. And that will happen also when there's a really high population of nematode in a bud. It's killed during the winter, and you don't have any leaves to see the next season. And you're going to have a whole lot of different effects on the trees. We know that they're reducing photosynthesis. They're causing that bud death that I was describing, and sometimes the leaves are going to drop off the trees earlier. That is often helped by anthracnose that can be in the population as well. These are some pictures that Amy Albom took in Blauvelt State Park in 1920. The 19 pictures are at the right, but I just wanted to make the point to you that, for example, if you look over here in 2020, one of the main things we see with this disease is that trees thin. And this happens in our ornamental trees as well as in the forest trees. The canopy gets thinner and thinner, and that affects even things like what grows on the forest floor because you've changed the light so much, which also changes the temperature. So we're having a lot of effects from the beech leaf disease everywhere it's known. This is the usual look that lets us know in the field that we're dealing with the problem. You can take these back to a lab and open up the areas and find nematodes within. And again, this is a one-time symptom. If you have 42 stripes on a leaf today, you're going to have 42 stripes on that same leaf in October. And it is a little bit different when you're looking at symptoms on the European beech, particularly since many of them have pigment in them besides green. And so there are times, such as at the left, when you can't really see the disease all that clearly. And I think people have thought of them as more resistant, partially because they can't visualize the symptoms as well. But in other cases, if you have the light penetrating through the leaf, for example, you can see some fairly impressive symptoms on European beech. And here's your Fagus sylvatica leaf that's showing very clear galls on the foliage. It's a little bit like peach leaf curl in its look, which is also a gall in foliage, but that's caused by a fungus instead of by a nematode. And at the right, you can see a thinning of the lower part of the canopy in this particular case. It doesn't necessarily happen in the lower part of the canopy first. I've seen other instances where it's in the top of the canopy. And again, here's a close look at the leaf tissue when it's got the infection. And you can go to a leaf like the one at the left there, and that one will enable you to lift up some of the gall tissue and look inside for the nematodes that are within. And here's one of the branches that we studied in our first investigations here in Long Island. Mina Becerra and I were looking at a study site in Wildwood Park, and we could very clearly see that even though the coloring would change over time from May at the left to October at the right, the actual number of galls was the same. And whatever you had in the spring was what you had in the fall. They have done some other things to look into what the impact of the disease is and how many nematodes are present. Looking at the buds is one of the methods. And you do want to look for the other kinds of symptoms as well. I think the most diagnostic symptom is the one where you see some discoloration between the veins. But there are times when I notice galls that are very flat. And when they're very flat, often they're sort of dead and not, they don't contain any moisture, and they're not really a site for the nematodes anymore. And that's the kind of thing I think we see in drier, hotter years. So that will reduce the population of nematodes for that year. We're going to have variation from one season to the next, according to how much it rains and how hot it gets and things along those lines. Here's some of the other things that you can do if you want to know about how the nematodes are perhaps moving off of one tree into the area around. You can put funnels out and catch the nematodes that are bouncing off the leaf when it rains. And that's one of the main ways we think that the nematodes move around. And then we talked about the bud checks, but there's also eDNA, meaning environmental DNA, now that we're able to really test for specific sequences of DNA that are characteristic of an organism. You can take samples from with an entire woodland and look to see where the nematode is and where it isn't, using that process without ever having to actually get a nematode by the tail and study it directly. We have a lot of work by DEC on this disease, which has been very helpful to us. They're very good at getting surveys going and learning where the problem is. And they educate the public on what they find and the importance of the trees as well. So we're talking about a state that we live in now that has beech, birch, and maple as the dominant forest type. And our beech trees are our fifth most abundant tree. And the birds and the black bears and the deer and the squirrels all really appreciate it for their nesting and for their food. These are some of the things that DEC did last year when they were working on beech leaf disease, and they've surveyed an incredible 27,000 beech, and almost half of the trees they surveyed were positive for the disease. And they're getting a lot of calls from the public. I'm very glad they're getting them instead of me. And last year they found 5 more counties in New York: Washington, Otsego, Oneida, Lewis, and Schenectady. They've started plots to measure how they might do forest culture in a way that would decrease the disease rather than making it easier for the disease. And that's an ongoing thing. You need a lot more than 1 year's data to understand the conclusions of those kinds of studies. But they'll do long-term monitoring to come up with those kinds of conclusions. And they have a couple of trials of ingredients that others weren't studying. They're looking at polyphosphite and chitosan, for example. And they're collaborating with various labs and, again, getting through to the public what's going on with this disease. They have a good website on beech leaf disease. This is the chitosan material that they were studying. This is one called Biosar. This is an example of something that's labeled for the disease control, but we don't have good data on its effect on disease control. In theory, it should be beneficial because it should have a general effect of increasing the plant's own natural defenses against any kind of disease problem that might be coming after it. But we'll just need to get more data before we understand the role of this particular kind of material against beech leaf disease. We just haven't had enough information yet. When DEC has looked at spread, it's been very interesting. You can see one little case here, for example. The white circle was the population of trees that was affected by beech leaf disease initially. The next year, the green area was involved. The next year, the blue area was involved. So you get a clear picture of spread of the disease from the initial fire that's lit. And it's just a radial moving outward, which fits with things like rain splash rather than a vector which might start more of the new fires. Whoops. Another place that's been very involved in studying the disease is the folks in Ohio, particularly at the Holden Arboretum. And they're doing molecular work and also field studies. And they do notice that they can find the DNA of the nematode sometimes before they can find the nematode itself. That requires an interpretation. It's hard to cope with that kind of information. And then they're looking at the effect on forest composition over time. So that's going to be very valuable information. And they're looking at the impact of the disease on the microbes in the forest as well as on the trees in the forest. So they're looking at a small scale and also a large scale as they put their information together. I mentioned the thin canopies. That's one of the main things that people have noticed. Some people go in there and cut trees down before they've died because they're concerned that they look really ill. And then some are actually going to die. Here in our area, I've almost always seen a second factor there as well. When I see death of a tree, there'll often be armillaria or a drought or something that seems to have been part of the reason why the tree died. So again, an arborist has a real advantage. They can try to provide really good cultural conditions for trees. And by providing those really good cultural conditions, they can prolong the life of the trees, even if they're not using a specific material to treat against the nematode. And that's just one of the benefits of IPM. Anything that you can do to irrigate trees in times of drought is very beneficial. Um, in the fertilizer category, some are phosphites. They're not actually a very good source of phosphorus, but they do have disease ameliorating effects. So those have been seen to reduce the disease in some studies. And then now we do have the option of using some fungicides, which we didn't have at the beginning. One of the most straightforward methods is to work with broadform, which has a 2 for use against this disease in New York. And it needs to be rotated with another material, so people tend to use phosphite fungicides as the thing that they rotate with. That's fairly convenient because many people are trying to combat Phytophthora problems on beech by using phosphite fungicides. So they can just then add in the broad form and rotate to get a legal kind of alternation of materials. And then we also now have the possibility of injecting with Arbotect, which is done as a macro injection every 2 years, and that became available to us in 2024.
Margie, I think that right now, since we're kind of transitioning over into some
Control.
control, let's pull up that second question.
Good timing.
So I opened it up. Have you seen any control with any of these New York State DEC registered treatments? Again, I'll remind everyone that if you're here for credit, you have to answer. If you're not here for credit and you want to, please feel free to go for that too. And one thing I haven't mentioned yet, if you have any questions, feel free to put them in the Q&A so that Marjorie can answer them at the end. But we got about 50% of people responding so far, so we'll give it another few seconds.
This is a question I wish we'd been asking every year, but it didn't occur to us because I think over time, people are, well, they started out as starry-eyed optimists, and then they got really depressed because it didn't go away right away. And now I think it's building back again to where more people are feeling that they're seeing some benefit to some of the things that they're trying to do.
All right, so let's end the poll and we'll share the results. 67% said no, and 17% were both at yes and maybe.
I think maybe is a good answer to that question. But I'll be showing you some things now that are a little more definitive than maybe, so that may change your notion. Although they're secondhand, they're not firsthand. We were sort of asking about your firsthand knowledge with that question. So there are some papers available in, this is in the Journal of Environmental Horticulture. There are a few other places, ISA, places where there has been some publication of studies of beech leaf disease, and they're really interesting. So you'll probably want to read some of these during the long, boring winter. And you'll learn something there about broadform's performance. It was brought into our attention because one of the ingredients in it is known to be a nematicide. Broadform isn't sold as a nematicide, but fluopyram is known to be a nematicide, and it's used in some of the other production systems. Trifloxystrobin is one of the strobilurins that we use for things like leaf spot control. So the combination of the two makes it an extremely effective fungicide. It's not even labeled on Long Island because they're concerned about its effects on organisms that live in water bodies. But it does have a 2EE registration for BLD in New York, and they are allowing its use just for beech leaf disease control here on Long Island. It started out as an 8 fluid ounce per 100-gallon rate, and now it's been reduced. Here are illustrations from one of the arborist group's trials. They have a nursery as well as an arboriculture business. The two trees at the left, which are looking rather lovely, were given a broadform spray back in the days when the 8-ounce rate was being used, and the 2 trees to the right were not given that broadform spray. And there's a real night and day difference. Again, you read the benefit of your treatment, not the same season, but you have to come back and look at the trees the next year. And that's the way you can find out whether you've been effective. So you definitely have to take pictures in year 1 to know whether things got better in year 2. Here's 2 trees from another experiment that I was able to witness in person. This is at Vertebra's Nursery on the North Fork in pretty much Aquabog. And this experiment was run by the Bartlett Tree Company. And the tree there on the, I'm sorry, my phone keeps going off. The tree there on the left was given a treatment with 6 ounces of broadform. And that was applied in rotation with phosphite, as I mentioned, so that you could get the opportunity to do more than one treatment with broadform. And the tree at the right was not treated, and you can see the difference in the 2 trees the next year. And then the exciting thing was that in this site, they treated at a series of rates. So if you look for the purpler trees, starting at the left, you have a 6 ounce per 100-gallon rate of broadform. The next good-looking tree only got 4 ounces per 100 gallons. The next one got 2 ounces, and then the next one got 1 ounce, and then you have another control tree that got none of the broadform. So there is a striking visual difference when broadform is applied to trees. It's not a particularly nice material to use in the environment, but it's very effective against this disease. So you weigh the advantages and the disadvantages, and you consider the location of the tree, and you figure out what's going on there. They took data on both the number of nematodes in the foliage as well as the amount of canopy visible or the amount of dieback of the canopy, whichever way you want to look at it. And the broadform had just extraordinary effects. At the left, you can see here, this is how many of the nematodes were there per gram of tissue, and almost nothing with all the broadform treatments, and then a really healthy number of, let's see, 8,000 per gram when you don't do anything. And we do know that the number of nematodes present is correlated with how much injury you see, which is why I think as we get more sophisticated with control of this disease, we're going to be able to look for treatments that suppress the disease. They won't necessarily have to take it to 0, but if we can suppress it, then we can keep the trees looking good. And we just need to get the finesse to understand how far we have to push it back to get that benefit. There's also been some research done in Ohio in the past few years looking at when to treat. And I think this is the other area for improved research in the future. A lot of folks have treated over and over and over again out of complete concern that they want to keep their beech trees. But here's the one study that looks at spring applications versus spring and summer versus summer alone versus the non-treated. And if you look at the numbers at the right, the first column talks about foliage. You have very little nematode in the foliage, very few of the nematodes, when you had a spring or a spring and summer or a summer treatment compared to the non-treated. So the difference is something between 0 and 4 nematodes versus 25 nematodes, basically. And then when you look at the nematodes in the bud tissue, it's the same sort of story. When you've done a treatment, whether it's spring, summer, or late summer, you're having something under 20 nematodes, whereas if there's no treatment at all, there's twice as many. So it's great to have some actual numbers so that we're not just telling each other what we think we saw. And I'm hoping there'll be more of this in the future, but at least we have enough to go on so we can go further with our experimentation. You might choose, for example, to try just spring treatments next year rather than treating later in the season. There are more nematodes later in the season. You can get that satisfaction of more dead bodies, but they're microscopic, so you can't see them anyway. So just having control of the disease is the important part. The conclusion of this most recent paper was that they don't think that something like a dense beech forest is ever going to get a benefit from fluropyram treatment, which is the broad form. And it would be too expensive, among other things, right? And it would be environmentally unsound as well. They also concluded that you can work with those lower than the label doses maybe not legally, but it does work. So you've got an extremely effective material that's probably not generating resistance very quickly because it's so effective. And then when you have those early summer or late spring applications, they may work even better than ones in late summer, which have been pretty much the standard thus far in the industry. Okay, so that's the broad form story. Then the one which has come on more recently is Arbotech 20S. And this one was known as an injection treatment for use on elm trees for Dutch elm disease for quite some time. And it's also been labeled for use on sycamore for anthracnose. It's just not as popular a use. And now this thiabendazole active ingredient is legal to inject into beech trees for BLD control. And there's a special local need labeling for that purpose that you're able to have in New York. The thiabendazole is a group 1 fungicide, and I just mentioned that to you because it's really known to have a problem with resistance development for whatever reason. I'm sure it's a very complicated reason, but we might find that we regret using this without alternating with another material, is my comment here. You've got a product that needs the 24C labeling to go with it to be able to do the beech treatment. And you're looking at an interesting formulation because the thiabendazole is in a hypophosphite formulation. And I think that might have something to do with the control, since we know that phosphite chemicals on their own are giving some benefit against the disease. So there may be some magic to the formulation that's used for injection into the trees. This is not used as a spray at all. It's just an injection. And this was registered for use in 2023. And it's very different from what we do with the broad form, which is just an outright spray. It requires fairly big equipment and a fairly large amount of time. You're looking at a gigantic tree here. It's 73 inches at breast height. And it takes a couple of people to do the treatment, and they may be there for 7 or 8 hours. And that is dependent upon the weather conditions and also whether the force is with you, I think. So it's not anyone's favorite kind of treatment to do, but if you're in an area where you can't spray the broad form, it gives you an opportunity for doing an injection that controls the disease because it's a very systemic material that moves well within the tree. So it can be quite effective. And you just have to cope with the amount of time you have to spend on a job and price accordingly. These are some illustrations from one of the Bartlett papers that hopefully you can make out. I think it's easiest to see here at the right. So you can see the banding on the top from a non-treated tree, and then the absence of banding below from a Arbotech injected tree the next season. And as you know, we have extra reasons to be a little nervous about injection because we're making wounds, and sometimes they heal very nicely, and sometimes they don't heal very nicely. And that's why the Arbotech injection has a very firm and fast rule that you can only do the treatment every 2 years. That should be effective, but we need to work with it and see if perhaps it could go to 3 years. It would be terrific if we could go longer. You have to use this treatment on trees that are 5 inches or greater in diameter, which is a little bit of a limitation, but not too much. It's usually the really large trees that are getting the extra attention of treatment for beech leaf disease. And it's important to work on the sides of the root flares. It has been effective. This is data from locations in Ohio, where you can see that in most cases when you did the Arbotech, you had less of a canopy contamination with BLD than you did when you had a non-treated canopy. And when they had a case where there wasn't as much difference, they were able to explain why that was coming out differently. Now we have one last thing, and I want to cover this with you because we've used phosphites somewhat illegally, I would say, for years because they can be applied to beech for the purpose of Phytophthora control, and people have seen some evidence that that also reduces the beech leaf disease. Some of them are sold as fertilizers. some are EPA registered as fungicides. The effect has been seen with both of those. These are, chemically speaking, mono and dipotassium salts of phosphorus acid, which doesn't mean anything to you, but if you want to know if materials are in that group, look for those words on the label. Here's an example of phytophthora canker that we control by doing a trunk spray with that kind of chemistry. But now we have a phosphojet material, which is coming to us just in time to have an alternative to the macro injection. So this gives us an option as of last year to do a micro injection of beech trees specifically for control of beech leaf disease, based on research that they've done in New England and on Long Island. And this is very different equipment. You have a small handheld unit that injects into the tree, and it's much quicker to treat an individual tree. The effect is visible, and I'll show you some pictures in just a minute, but you can see the amount of equipment is extremely different, and the volume is very different. Here you're looking at a non-treated tree in the distance, and the two green ones on either side are two of the ones that were treated using the phosphojet. And here you have a green tree on the left that was given the phosphojet treatment, and the tree to the right of it was not given it. And here's some of their data. In Jamesport, at the nursery that I visited, they were looking at an experimental as well as looking at the phosphojet, and they also looked at the combination of the two. And if you look at the far right, you'll see the appearance of the trees in 2025 after they treated in 2024, at which time they were all very similar. And you can see that there is a real reduction from, let's say, about a 40% amount of symptoms in your non-treated check down to almost no symptoms or lower symptoms when this phosphojet was used. How often this would be used is an interesting question. I again go back to the idea that maybe we're going to be able to just push it back and then come back a few years later to do another treatment rather than having to treat trees every year. Okay. It wasn't exactly what I intended to do, but I think that will be fine. So let's take some questions. I just want
All
to make
right.
one more
Well,
point with you.
while you do that, I'll put
Mhm.
up the next poll,
Mhm.
the last poll.
Please do.
So you So it's have you observed any trees that appear to have natural resistance? Yes or no? I guess maybe is another one I should have thrown in for this one, but
hmm Yeah, we wouldn't expect you to know scientifically because I haven't heard of any official reports of resistant trees. But I'm just curious about your observations.
so, and if you are here for credit, make sure you're answering. All right, we'll give it another few seconds. We got some yeses,
hmm That's
which is
good.
always interesting. Okay, so I will end the poll and share the results. So we got 27% yes and 73% said no.
That's very interesting. Okay. So the 27% of you who think you may have seen resistance, keep watching those trees. I did want to make an appeal to you that I think it's very important for us to find resistant trees. The disease has been out in the natural population and the ornamentals population for quite some time. So among the ornamentals, we might be able to identify cultivars that are relatively less prone to the problem, particularly among those European beech. And then in the woodlands, we have so many trees, we might just plain find some that aren't as susceptible, and then we could propagate from those. So please do watch for a while, and when you become convinced, contact the DEC or Cornell or some authority who could help you decide if there's any validity to the resistance of that tree, and that would be just terrific. Okay, so that's what I have for tonight. Anyone else have questions?
So we have one question. What is the mortality rate for the, oh, we have a couple. What is the mortality rate for the disease? Did you say large trees may tolerate it?
Large trees may tolerate it if conditions are good. There are reports from Ohio of 10% of the large trees dying in the forest in the first, let's see, 20, first 10 years, let's say, of the disease being present. I haven't seen an updated figure on that, but there are trees that are dying, but not every tree dies, and we do need to find which ones aren't dying and make sure they've been exposed to the problem so that it's not just escapism. And that's real important. The younger trees are more susceptible, so if you're just a little sapling or you're a root sprout from another tree, you're much more likely to die because you don't have the resources. You don't have the root reserves, the starches to make it through a year when you can't photosynthesize as well.
Awesome. And I will just real fast say, if you're here for credit, it is 7 o'clock. Thank you all for coming, and thank you, Marjorie, for being here. If you are here for credit, feel free to put in your name and your license number in the chat. And you know, I hope everyone has a wonderful night. But we do have one more question, so anyone
Okay,
who
yeah.
wants to stick around, feel free to.
Mhm.
Keith said, I do not see beech nuts on infected trees. Is that
Mhm.
common?
That's very interesting. I don't know much about beech nuts, but I can tell you that at Bayard Cutting Arboretum, we have seen beech nuts on some of the affected trees, but it's not on all the trees. The conspicuous beech nuts are on certain trees. So I think that trait may vary a lot from one plant to another. Maybe it's one of the variations within the species is how much um, seed production there is. And if it's correlated, that would be really interesting. So test that theory as you go out and watch beech trees. Maybe the ones with the disease are just not in a good mood, and so they don't want a fruit. I don't know. So I think that's all we have, right?
Yep. Now we just have
Okay.
some thank yous coming in.
Oh, great. Okay, that's
Yeah.
nice.